So thank you so much. First of all, I would like to thank the sponsors for sponsoring this wonderful session, and also to the organisers for really making this possible. Getting the invitation to come here was something very special for us. And as a previous president of the Australian Society for Parasitology, I'd like to pass on best wishes to both of the societies. So I think, and particularly thank you to Russ and Bonnie for sort of hosting me and supporting my trip across. But it was a great opportunity really to ambush David because he has been an extraordinary sort of mentor and supporter of our, not only myself but also our team. And we are very grateful to your support over the years. It's been very collegial, philanthropic. It just works, and so we sort of get things done. And it's been a great pleasure. So thank you very much. So this is so David was sort of giving the original title. I've modified the title. It's a bit crazy, but I suppose I'm a bit crazy too, so it doesn't really matter. Expanding the genome universe for parasites has been something that I've been really interested in for quite some time now, and not because we're making, sort of solving all of the problems in genomics, but because it's raising a lot of new questions as well. But what I wanted to do today was really give you a bit of an update on progress in genomics, not only of parasites, but also going back in history a little, just to give you the context and show you why this has been so exciting, at least to me. So it's a bit of a personal perspective, and I'd also like to look towards the future and look at the challenges and opportunities in this particular area. So when I was a vet and started out in research in parasitology, I knew absolutely nothing about how much impact some of these technologies were having. But as we know now, some of these techniques have really changed some fields, not only in parasitology, but more importantly in other areas. It always takes some time for these techniques and technologies to really penetrate our field of parasitology. But if we think about just PCR as an example, the technique that Carey Mullis invented in 1985, that was a huge breakthrough for all of us in terms of being able to analyse genes, amplify them from small amounts of material. Without PCR, we'd be lost in parasitology. And also the developments in, developing the GenBank and having that database available to deposit material, but also retrieve material and store material has been a massive advance for us. But it's really the advent of automated DNA sequencing will really change things forever. And, we go back to sort of the early 1990s where things started to happen using Sanger sequencing. This is the automated version of sequencing. And really this was the beginning of a revolution, if you like, in addition to the fact that databases were being produced that were specific to certain areas, but also the general database, NCBI, and the access to the internet, World Wide Web, just made it possible to share huge amounts of data across the globe, which just hadn't been possible before. And I remember very clearly when we for the first time, dialed into the World Wide Web via modem. Actually, initially, it was the start of a revolution. And if we think back, it wasn't that long ago that the first metazoan organism had been sequenced and its genome assembled and annotated, and that was Caenorhabditis elegans, the free-living nematode that Sidney Brenner was working on for years and years. So in the 60s he isolated the worm from soil and then cultured it, and that became a model organism that was used in many labs around the world to try and understand the biology of metazoan organisms. But at that time we didn't have the genome, but in 1998, when the genome became available, that was really the starting point for us to understand the fundamental aspects of genes, how they work, and try and understand the connection between the genome and the phenome of the organism. And it wasn't only the basis for the worm itself, but it was also the basis to understand generally in metazoan organisms how genes work, including vertebrates, including humans. So there's a lot of studies that were done in C. elegans that relate to genes from humans. And so that was 1998, but then in 2000, Drosophila genome was sequenced and published, and that also provided another perspective. So looking at both organisms and cross-comparing them also gave us a better understanding of metazoan organisms and eukaryotes in general. And it wasn't really until 2001 that the human genome was sequenced, and in 2003 the curated version of that genome was made publicly available. And so really, if we think about these model organisms, at that time, the integration of genomic data into the worm-based database that Paul Sternberg had initiated, as well as the Drosophila fly base by Bill Gelbart, that was really the start of, you know, the sort of integration of genomic information with biological data. And that was very powerful because then it gave us insight into a lot of areas of organismal development, reproduction, neurobiology, all of these issues, cell differentiation and so on. That really was a starting point to understand, really at the functional level, these organisms and draw inferences in relation to other, you know, higher organisms such as humans. So, but the next major breakthrough, of course, was, you know, was the book of life, sequencing the human genome, having the code in a book, but not really understanding all of the language that went with that code. And still today, there are many areas that we don't understand in relation to gene function in human genes and so on. So there's a lot still to be learnt, and this relates to the fact, I suppose, that we still know very little about the functionality, and this goes back to, I suppose, some of the comments that I'll make toward the end of the talk. So the cost of the human genome was somewhere in the order of $3 billion, and when the information became available, some scientists were pretty disappointed because originally they'd predicted about 100,000 genes, but it ended up being about 22,000 genes, and those 22,000 protein encoding genes were only encoded in 1.5% of the whole genome, which was astonishing. So a lot of this genomic information was not understood. And so, but irrespective of this, you know, these developments through C. elegans, Drosophila and human genomes then really opened up a revolution in genomics. And this was, we were also part of that in parasitology. We were part of that, but certainly it expanded into all areas of genomics, and there were many genome projects initiated between 2004 and 2010 and beyond, of course. But it was really this genome of Brugia malay that stuck out because it was one of the first ones in 2007, but it was also one that was still today is a very well assembled genome, very well annotated, and that was because the original strategy of using bacterial artificial chromosomes was used to undertake the sequencing, so that we had longer stretches of DNA being sequenced in one piece. And so this was really a spark for the parasitology community to get stuck into sequencing some genomes, and it was a spark also for us. So it was Sanger Institute was in, and the McDonald Institute in the States was also in there starting to sequence genomes. But really our mission was really to try and understand parasites better at the molecular level through genomic information, and then also understand the relationship between the parasites and the host animal. and then the disease processes using, some of these advanced technologies, with a focus, of course, at trying to develop, new intervention strategies and diagnostics for the future. So gaining good information through a global understanding of genomes was really our focus, and we were, sort of considering different parasites, but our main focus was really on selected examples of parasites that cause neglected tropical diseases, and also some that were of animal health importance, particularly those that were transmissible to humans. And the reason for us focusing on this area of genomics was really to try and underpin future development of future intervention strategies. And this was needed because obviously a lot of these parasites are quite hard to control. Some of the problems that we see are, there's no vaccines available for quite a lot of them. There's drug resistance problems in parasite populations, particularly those of livestock, and problems with diagnosis as well, and even toxicity of certain drugs on the host. And so there was a need to try and get a better understanding of parasites at the molecular level. So in 2009, we initiated some discussions with Beijing Genomics Institute in Shenzhen in China, and initiated some pilot projects using transcriptomic approaches to try and look at different genes expressed at certain time points in the life cycle of particular parasites, including Haemonchus and some flatworms. And then eventually we got funding from the Australian Research Council and National Health Medical Research Council to then undertake real genomic sequencing. So when I got back from sabbatical at Caltech with Paul Sternberg, we started our sequencing effort. And this was sort of just after the panda genome sequence was published, and this was really the start of a revolution, because at that time Illumina technology became available, and you could use a single technology to then sequence the genome of an organism rather than having to clone DNA into vectors prior to sequencing. So this was a really, really massive breakthrough, and of course, together with the computational approaches that were being developed and the availability of worm-based and fly-based, this really opened up the field. And so, given the complexities and idiosyncrasies of parasite genomes, we also had to, from scratch, develop some bioinformatic tools to allow us to, number one, assemble genomes properly, but also then to annotate them properly and move forward with further analyses post-assembly. And so we published a number of papers in the field and looked at various genomes to get a global impression of the molecular landscape of some of these beasts. But then we also dug deep to try and understand some of the aspects of biology. And I suppose, given the time constraints, I can't give you deep insights to all of them, but I will give you one example, and I'm taking a veterinary example just to please Joseph, using Haemonchus as an example. As many of you know, Haemonchus contortus is really a parasite of huge importance in the veterinary field because it causes losses, particularly in the subtropical and tropical areas. It feeds on blood from the stomach wall, and there's widespread losses as a consequence of acute deaths, but also chronic disease. And there's also drug resistance problems because a lot of these drugs are widely used around the globe, anthelmintics, and this has led to major issues to do with drug resistance. And so we wanted to really delve deep into the parasite and sequence its genome. And it took us 3 years to assemble the genome of Haemonchus, and I won't go into the details, but there are some suspicions that this thing is actually polyploid and not diploid, and so there's new information that indicates that it's a very complex story biologically. But we really busted a gut to try and get a genome through various rounds of pre-processing of the data, and really the upshot of it was that it was very difficult to assemble because of the sequence variation that you see in genes across the genome. But nonetheless, we succeeded, and we produced a draft assembly that was in the order of 320 million bases, with about 25,500 genes at the time, 23.5, sorry, and eventually getting an assembly that was moderate, but not what we would call top quality sort of assembly. But nonetheless, we were able to work with that, and we were able to then annotate it and work towards understanding the biology of the parasite. So what we did was to look into certain aspects of the way that genes are transcribed in the parasite. This parasite has a direct transmission pattern similar to, let's say, a hookworm, although this is, in this case, it's truly oral transmission. So you've got in the host component and free living stage or phase of the life cycle, and as these eggs go through to development of L1s, you get a massive upregulation in genes, see a reduction as the parasite adapts to the environment. You see a reduction in the number of genes that are upregulated, but then another further reduction as it goes through to the L3 stage, which then becomes essentially a motile stage, but is akin to a sort of arrested stage, if you like, in the sense that it is still motile, but it doesn't undergo any further development. And so once this is ingested, of course, and exposed to host juices as it goes through the gastrointestinal tract, there's a massive upregulation of molecules that the beast needs to survive and gain access to nutrients, obviously a lot of enzymes, ES products and so on. And eventually you'll see the differential expression of genes that relate to reproduction in males and females. In addition to that, we were able to look at molecules that are involved in parasite-host crosstalk. What we mean here really is the secretory molecules that are expressed or transcribed in this case by the genes, and you can see that a large proportion relate to peptidase and peptidase inhibitors. And if you look at the breakdown of the peptidases, you can see that metalloproteases and aspartic proteases, cysteine proteases, make up a large proportion. A lot of these play roles in immunomodulation, immunosuppression, and even activation. And some of them indeed have been used as vaccines. And currently is this HGAL-GP complex, which has been identified, of course, has been worked on for years, is protective in sheep against Haemonchus infection, which is, and so a lot of information just through the transcriptomic analysis that we've done. In addition to that, we could focus on identifying and predicting essential genes that relate to key biological functions that are really crucial for the survival of the parasite. And we do this by inference, by comparing against Caenorhabditis elegans and Drosophila. And we identify these genes, and essentially what they are, or what they represent, is potential drug targets, because the proteins that are expressed are essential, and those proteins potentially can match up with chemicals in databases. And so what we've been doing is identifying chemicals that match these sorts of molecules to prioritise them and then test them in a phenotypic screen system that we've set up for Haemonchus. So we're actually prioritising chemicals and then screening on whole parasites. In contrast to the systems that have been used in industry, which are mainly focused on using the free living stage of nematodes, we're using the parasitic stages. So we activate, we exsheath L3 stages, activate those and get them through into the L4 stage, and we measure different aspects. So we measure motility of the third stage larva. We also measure the motility of L4s and also the development of the parasite in vitro. And this has given us a beautiful tool to screen quite large numbers of compounds, and we're working together with Medicines for Malaria Venture now at the moment, looking at identifying particular lead candidates, and we've got a number of them that we're now working through this system to optimise the chemistry of these compounds and then work towards assessing you know, the absorption, distribution, metabolism, excretion, toxicity, as well as in vivo testing of these compounds in animals. And so we focused on using Haemonchus as a model system, but we're also mindful that we need to focus on trying to provide any lead candidates that we have to test on human parasites as well. So this is just a really brief example of, you know, the Haemonchus story. But really, if you look at the current state of affairs now with parasite genomes, there's about 100 nematode genomes in worm base. There's about 33 flatworms. There are some nice little reviews that you can, if you're interested, look at. But one thing that's come out of all of this is that although we've got this genomic information, there's still quite a lot of gaps in our knowledge, particularly in relation to orphan genes. So these are genes that we know nothing about. They have no homologues in current databases. They seem to represent parasite-specific molecules, and they are particularly exciting to pursue, but they're hard to work on. And so we're looking at trying to tap into this information to look at this essentially gold mine of molecules to see whether any of those are essential targets. And it's interesting to note that if you look at the genomes, the genes that are known from parasites, there's only 10 to 50% of the genome codes for these sorts of protein genes. And so there's a huge amount of the genome that really is unknown, and we don't know what its function is in most parasites. And in the early days, this used to be referred to as junk DNA. And, you know, junk DNA is a term that people used to use. Maybe perhaps today we use non-coding DNA as a more correct terminology. But it's interesting to note that if we look at parasites, they're more akin to, let's say, vertebrates, in that there's a lot of genome that's unknown, and only 2%, for example, in humans, 1.5 to 2% of the genome codes for protein-coding genes. But in the pufferfish, 98% of the genome codes for protein-coding genes, and only 2% of the genome is junk DNA or non-coding genome. So it's just useful, I think, to just remind ourselves that if we look at the DNA level, genes are organised in this way with exons and introns. RNA is transcribed, and the pre-RNA contains these elements. But then eventually it's spliced. This is chopped out, the introns. These introns represent basically this junk DNA. And for years and years, we knew nothing about this field. But there was a guy that I got to know quite well, Malcolm Simons, up on the left. And Malcolm was a medical doctor. And quite early he saw that given this large amount of information in the genome, there must be some function related to the junk DNA. And later on, as things became clearer, they patented junk DNA in prophetic patents for all living organisms on the planet, surprisingly, and actually held these patents for many, many years, but they've run out now. But eventually, as it became clearer that this junk DNA had function, John Maddox stated that this was probably one of the biggest mistakes or failures in the history of molecular biology, not to recognise that junk DNA was really crucial in regulating a lot of biological functions. So junk DNA really encodes promoter elements, long non-coding RNAs, short inhibitory RNAs and microRNAs, and all of these things will play a role in parasites, but we don't know much about these aspects in parasites because we don't have any model systems. We need to remind ourselves that out of this long strand of DNA, it's about 1.5 to 2 metres in length, is packaged into a nucleus which is only about 5 microns in size. But there's all sorts of interactions going on between DNA and RNA, and DNA as well as protein and so on. And these interactions determine this is essentially a 3-dimensional dynamic structure in which there's a lot of regulation going on all the time. And it's a dynamic structure. And it wasn't really until the Mod Encode project was developed, that we started getting insights into the different functions of non-coding DNA in Caenorhabditis elegans and in Drosophila, and this is now helping us understand how these genomes are regulated. And there are a lot of external factors here that influence how this works and induce changes in the regulation of the genome and the genes, and we could conceive that a lot of this regulation has sort of underpins some of these key biological aspects that we've been so concerned about: hypobiosis, synchronous emergence of cythostome larvae, for example, and developmental transitions. So there's a lot going on. So in conclusion, I think we've made a lot of progress in the genome area, but there's still a lot of draft genomes available, and so we really need to finish those genomes, work towards solving some of these challenges to do with functional genomic platforms, bioinformatic bottlenecks, and work towards applied outcomes with protein encoding genes that we already have available. So there's a lot of need for capacity building in the area in parasitology, because there's still a huge bottleneck in the bioinformatics area. And I think it's important to just remember that there's a lot of complexity around this aspect of dark matter in genomes that we know virtually nothing about, and could provide, if we understand it, could provide a window to understanding parasites, their interactions with the host and disease, but also potentially underpin radically new strategies to combat parasitic diseases. So thank you very much for your attention, and again, thanks to the organising committee for inviting me. Thank you.